Hello, my name is Cindy Lopez, and today I will be presenting the molecular mechanisms of the sliding filament model. Um, so before I get started, I just wanted to kind of introduce to you guys what a sarcomere is. Sarcomeres are found normally in striated um, muscles, such as the skeletal muscles and cardiac muscles. And then they consist of basically thick and thin filaments, as indicated here, and how they're arranged is an A band is where the overlap of the thick and thin filaments are, with the H zone in the middle, and an M line. The Z lines are basically going to be the length of the single sarcomere, and where there isn't any thick filament present, that is considered the I band. Just to kind of go a little bit more into inside of what a thick filament is, it is approximately 15 nanometers in diameter and 1.6 micrometers long. It is made of myosin, which is really important, and they lie parallel in the middle of the sarcomere. So where that bare zone is, basically what's going to end up happening is that M line is going to be running down the middle there. Globular domains protrude from the thick filament, so those weird little bumps that you guys can see are going to be the globular domains, and globular domains are super important because they allow the formation of cross bridges between thick and thin filaments. Thin filaments, so they're approximately 7 nanometers in diameter and 1 micrometer long. They're made of actin, mostly, tropomyosin, and troponin. They're all kind of important. Troponin is going to be a complex that is made of troponin T, troponin C, and troponin I. I believe troponin T means troponin tropomyosin. Troponin C, or sorry, TNC is troponin calcium, and TNI is troponin inhibitor. And before I move on, I just want you guys to kind of see how these thin filaments are arranged. So you have the actin, or in this case the F-actin, that kind of runs parallel with the tropomyosin, kind of running along the grooves of the actin molecules, and then you have the troponin complex that are going to be kind of everywhere in the thin filaments. So here is the sliding filament model. So when you're looking at it, especially under a microscope, right, you see that the A band, as it contracts, can remain fixed in length, and the I bands tend to shorten. So you can look at the image here, and you can see exactly what I'm talking about. But what actually happens is that those bands don't necessarily change, or that I band doesn't shorten. It just like kind of runs into itself in between the thin filaments. So what actually happens is the thick and thin filaments pass against each other, and there's no change in length in either of the filaments. Here I'm going to explain the process of muscle contraction. So we're going to start at the high energy cocked configuration of the globular domain that contains ADP and P1 molecules. After that, there is a transition of myosin that triggers a conformational change that then releases PI. This then again makes another conformational change called a power stroke, where the thick filament pulls against the thin filament, and then from that interaction, ADP is released. Afterwards, the myosin chain can stay attached to the filament, to the thin filament, I should say. A cross bridge is disassociated and would need ATP to bind to the myosin. So basically, what ends up happening is that the ATP will come in and detach myosin from the actin. The myosin chain then changes conformation again, which weakens the binding strength within actin and then detaches. And you can see that in this middle image here between 5 and 6. And then finally, step 6, ATP hydrolysis returns myosin chains to high energy configuration and is basically ready for contraction to begin again. And then in this last slide here, I just want to emphasize on the importance of calcium in this whole process of the contraction, right? So muscle contraction heavily depends on calcium availability. What normally happens is that tryptomyosin blocks, so it's an inhibitor, right? It blocks the myosin binding sites, but depending on the amount of calcium that is present in the environment, or the calcium ions that are present in the environment, would allow the troponin C, which is part of that complex that I mentioned earlier, to bind to the ions, which then undergoes another conformational change, and then it'll be transmitted to the tropomyosin, and then the tropomyosin no longer blocks the myosin. So this is just for one globular unit that I mentioned before. When you look at, like, for example, a video that's explaining on how it actually works, there's all these globular complexes, I guess, that are undergoing this action here, and so when they're binding and unbinding, you're having multiple different reactions happening, and that's basically that pulling of the thick and thin filaments. All of this is just happening at that point. Thank you so much for listening to my presentation, and these are the references that I gather my information from. I'll see you guys in the next one.